Let's see the animated version of Griffith's experiment for better understanding. Frederick Griffith performed a series of experiments to prove that DNA is a genetic material. In his first experiment, he injected the live S3 strain into the mice died. In the second experiment, he injected the live R2 strain into the mice survived. This is because the live R2 strain was non-capsulated and it was non-pathogenic. In the third experiment, he injected the heat-killed S3 strain into the mice and the mice survived. In the fourth experiment, he took the mixture of the heat-killed S3 strain and the live R2 strain and he injected this mixture into the mice. Unfortunately, the mice died. So this brought a confusion in Griffith and he gave a conclusion saying that some material in the live R2 strain have transformed the heat-killed S3 strain into live S3. In a previous video, we have studied about the Griffith's experiment of transformation, where we have studied the 4 different experiments performed by Griffith, where in his final 4th experiment, he found the unusual result and he could not come with a proper conclusion. Whereas in this video, we are going to see the 3 different scientists after 16 years, they took up the same experiment of Griffith which laid as a foundation. Now we know that in the 4th experiment the live R2 strain was injected with the heat killed S3 strain into the mice and the mice unfortunately died. This and Griffith concluded saying that some material in R2 strain were transformed the live S3s into the live S3 strain, whereas after 16 years after performing the Griffith experiment, 3 scientists, Avery, Loed and Mc Carty, performed the experiment taking the Griffith experiment as a foundation to prove that DNA is a genetic material. Now they took the tissue of the dead mice and they analyzed. They found that the tissue contained live S3 strain of Streptococcus pneumoniae. Seeing this, they came to a conclusion saying that some material in the S3 strain transformed the live R2 strain into live S3 strain. They concluded saying that some material in the heat-killed S3 strain transformed live R2 strain into live S3 strain. It may be any biological material such as protein, DNA or RNA. Before, it was believed that protein to be a genetic material, whereas after Avery, Loed and Mc Carty performed a series of experiments taking 3 enzymes. They took 3 enzymes. First, protease. Protease is the enzyme which was used to degrade proteins. When this enzyme was used, the mice again died. The same result was found. They took RNase enzyme which used to degrade RNA and the same result was found, and this proved that RNA is not the genetic material. Again he took the third enzyme, DNase. Taking DNase for the experiment, they found that the mice survived. This because the DNA got completely destroyed. Once the DNA got completely destroyed, it was proved that DNA was the genetic material. The conclusion of the experiment is that the DNA, the genetic material of the heat-killed S3 strain, transformed live R2 strain into live S3 strain, which made the non-pathogenic live R2 strain to become pathogenic that is live S3 strain. The experiment conducted by Avery Loed and Mc Carty was most welcomed by the scientific community, and this proved that DNA is the genetic material.